Well, hi everybody, and good afternoon, and welcome to our monthly Envy Smart meeting. My name's Brad Clark, and I'm an academic from the University of Melbourne. I would like to acknowledge the traditional owners of this country, wherever you may be today, and pay our respects to their elders, past, present, and emerging. Now, to our keynote today. It's one of the great things about Envy Smart is we get to move across a wide variety of different topics. We've had some waste management talks, we've had soil chemistry talks, and today we're going to learn about radioisotopes. And we have Dr. Tom Cresswell joining us. He's an ecotoxicologist, and he's passionate about understanding how humans impact aquatic ecosystems. He has a background in marine science from the University of Plymouth, which includes marine biology, chemistry, and physical oceanography, as well as subjects in environmental economics and maritime law. Tom completed his PhD in ecotoxicology and environmental chemistry in 2012 with RMIT and CSIRO, which saw him work on a river in Papua New Guinea downstream of a gold mine. He's been working at ANSTO, Australia's nuclear science and technology organisation, for the past 9 years on the development of nuclear science in ecotoxicology. Welcome, Tom, and thanks for being here today to give the keynote.
Thanks very much, Brad, and thanks everybody as well. I'd like to acknowledge the Darawal Nation, where I'm speaking to you from, and acknowledge elders past, present, and emerging from that country. Brad, can you just confirm that you've got full screen of my PowerPoint?
Tom, I can confirm that I can hear you and I can see you. And also, maybe at this point, I should encourage the participants, get armed with some good questions for Tom when he finishes, so we can have a good discussion at the end of this. So you're all set, Tom?
Please do. Thanks, Brad. And I promise to be to time as well. Brad got a little bit concerned when he saw the number of slides, but hopefully I can get through this without going over time, but please do jump in. Thanks for that introduction. As Brad said, I'm an environmental toxicologist at ANSTO. We're at the Lucas Heights facility in southern Sydney, which is home to Australia's only nuclear reactor, our research reactor Opal. And it's through that reactor that we generate a lot of radionuclides that you'll see used in the presentation here, and hopefully I can convince you that nuclear techniques can be good in terms of use in environmental toxicology and environmental science. So let's do this. There we go. So we're all aware of the benefits, the economic benefits and societal benefits of growth, of urban development, and industrial development, and what it can do, especially for the economy. But obviously over the last couple of decades, we've become very aware as to the potential impacts and stresses that it can cause on our environment. And in particular, my interest is in the aquatic environment, all the way from fresh waters through to open oceans. And as you can see from the pictures here, a lot of what we do as humans can impact our environment in ways that we're understanding very well, but obviously we're still understanding, there's still a lot more to understand of those impacts, especially in such dynamic systems as aquatic environments. So in general, in ecotoxicology, we ask that question of what effects are chemicals and other stresses having on animals and plants in the receiving environments, and why is this why do we ask this question? Well, by protecting all the way from an individual organism through to populations, we conserve biodiversity. And in doing so, that allows us to maintain clean water, clean air, and food. So it's always a good question as to why conserve biodiversity and look after our environment, because that provides us with the three things that we really need to maintain ourselves as well as everything else on the planet. When it comes to monitoring compounds, contaminants, chemicals in the environment, we've got very good at water quality monitoring, and we as the community can accurately determine concentrations of compounds that are very, very small within the environment to amazing accuracy. But often this kind of sampling just provides us with a snapshot in time and not how those contaminants are changing, unless you're going back and doing lots of sampling over time. Biological monitoring, on the other hand, provides us a bit more time integrated, as the animals and plants living in the water will integrate that contaminant loading over time. But for mobile animals, fish, invertebrates that move around, it's very difficult then to, when you've pulled an animal out of the water, to then determine how long it's been there in that water and what that exposure history is like. So when we're trying to understand impacts to aquatic ecosystems and especially bioaccumulation of contaminants to do impact assessments, we're asking these types of questions. So what's the predominant source of the contaminant to the organism? Will the contaminant be retained by the animal or by the plant, or will it actually, is the animal and plant able to release that back to the environment? And where does the contaminant end up within the animal, i.e. what's that target organ that may cause stress? So it may be that it's the contaminant is going to the reproductive organs, and so you've got issues with future generations, or potentially it's something to do with growth. So understanding and trying to answer these 3 points is really key. And again, us as a community of ecotoxicologists and environmental chemists have got some really good answers to a lot of these questions. And they've come about traditionally through conducting a series of exposures. I should have made a warning here for anyone squeamish. In fact, I don't have it up there on the screen. So if anyone's squeamish, please look away. But the point here is that in order to understand how quickly contaminants are uptaken in an animal or lost from an animal, we will often try to construct curves like this. In which case you'd have multiple animals exposed over time. You remove a subset of those animals. In the case of metals, you digest them in acid and analyze them by ICP-MS or ICP-AES. You require a lot of animals to do this, and you're making assumptions that the animals at the previous and following stages are all behaving along that same line as well, in order to generate these kind of time versus concentration curves. And again, for the squeamish, look away, but for those that are not, this is traditionally how we would do organ distribution as well, to understand where the contaminants are within the body. Is room temperature dissection. And if you've got mobile contaminants that are potentially sensitive to dissolution, then there's potential here to cross-contaminate organs or lose any of the contaminant from that organ in terms of just the bodily fluid. So there's potential issues with this, but obviously it has worked pretty well in the past, and what we try to do with some nuclear techniques is to advance that. So using nuclear techniques, we can use fewer animals, which is always good for the 3 R's in terms of reducing the number of animals used for research, especially good as well for animal ethics committee concerns. We can also use these techniques to understand differences among individuals and real-time biological processes, which I'll show you some of the examples of that, and then also determine where the toxicants end up within an animal, which gives us some information about the toxicity. Going back a little step about using radioisotope tracers and nuclear techniques, what are they? Well, a radioisotope is a radioactive form of an element. So in this case, we've got the 3 isotopes of carbon. Carbon-12 and carbon-13 are stable. They're not radioactive, but carbon-14 is radioactive. And we use that one a lot for carbon dating, because it is radioactive and we know its half-life very well. And the key for doing radiotracing is that chemically and biologically, the radioactive form of a chemical behaves exactly the same as the stable element, but they give off a radioactive signal which allows us to detect it. And these signals can come in the form of alpha radiation, beta radiation, and gamma radiation. And it's really the gamma radiation that allows us to be able to detect what's in an animal while it's still alive. So how does this work in practice in the lab? Let's say we're looking at cadmium, selenium, and zinc. We take the gamma and beta emitting radioisotopes of those metals, add it in this case into solution, and here we have a tadpole in solution in our exposure. After a certain period of time, we remove the animal to remove any and then rinse it to remove any loosely bound metals that are on the skin. And then we put it into a smallish container, but still making sure it's got enough water and enough dissolved oxygen, but a container to restrict its movement. And because it's giving off a gamma radiation signal, we can place it on a gamma spectrometer shielded from external radiation. And that allows us to work out the activity, how much radioactivity is within the animal while it's still alive. So very similar to a CT scan if you go into a hospital to do that sort of thing. So on a whole body basis, we can then quantify how much of each of the radioisotopes is present within the animal. And then we can convert that into a mass of metal so that we work out, okay, it's got this concentration of cadmium, selenium, and zinc at that time. And then because the animal's alive, we can return it to the exposure, rinse, repeat, and we can come back multiple times so that we're getting an individual's response to the uptake of a particular contaminant. And this allows us to generate curves like this. So these are time on the, almost all of my plots are time versus concentration, time on the x-axis and concentration of a contaminant on the y-axis. In this case, this is freshwater prawns response to dissolved cadmium. And I mentioned that we're able to see things like biological processes. So where the triangles here on the plots, you can see that this is actually a molting event. So where the animal has shed its exoskeleton, it's then had a brief but significant uptake of cadmium before returning to a normal uptake rate. So there's, in this case, we believe that the animal is trying to pull more calcium from the surrounding water in order to ossify, harden its shell, and in doing so is inadvertently pulling cadmium in and bioaccumulating cadmium at a greater rate from the surrounding water. The other thing that it allows us to do is use the accompanying beta radiation that is often there with the radioisotopes that we use to look at organ distribution. So we take the animals that have been exposed, we euthanize them humanely, embed them in a resin, and then snap freeze them in liquid nitrogen and section them frozen on a cryomicrotome. And then we take those tissue sections and expose them to a phosphor plate, which allows us to image where the radiation and therefore where the metal is within the body. So in this case, we've got an identification that the cadmium in this particular example goes to the hepatopancreas, so the prawn's equivalent of a liver, and the gills being the site of uptake of this aqueous, this dissolved cadmium exposure. So it really gives us a really nice insight on a whole body perspective as to where metals are. And we can use these imaging techniques semi-quantitatively as well to say that there is X times more in one organ than there is in another organ. So I'm going to jump through a few case studies. I've got 7 up here on the slide. We'll see how far we go through. I'm keeping an eye on the time, but just to give you a bit of a flavor of the different ways that we've used radio tracing in terms of aquatic organism studies. We have also done some work with terrestrial plants, crop plants, understanding things like phosphorus or zinc as macro and micronutrients uptake into different plants, but that's probably for another conversation. Otherwise, I'll go way over time. So just running through these, how we've used radio tracers in our labs over the last few years. The first study is on understanding the kinetics of bioaccumulation, retention, and internal distribution of radionuclides. So this is really understanding what happens following Fukushima, where you have a significant release of radionuclides to the marine environment. And in this case, we took a model organism for looking at radioactive doses. So in the marine environment, that model invertebrate is a crab. And so we exposed the crab to a series of aqueous and dietary and sediment exposures of cesium and strontium, identified biokinetics, and then also looked at organ distributions as well. And by combining both the biokinetics, so the rates of uptake and loss, as well as internal imaging of where the radionuclides went internally, it's given us some better predictions of radioisotope transfer to organisms and dose estimates following some of these catastrophic events that hopefully don't happen very frequently at all, but yeah, still enable us to be a bit better at predicting potential effects. So one of the first things that we want to do is to understand which is the predominant source of uptake to a particular animal. And you can do this in a whole range of freshwater or marine organisms using these techniques. And this is coming from some work done by Lundrum et al. In 1992. And it's described by a steady state biokinetic model. So the concentration within the animal, CSS, at steady state, is described by the uptake from water, so via respiration across the gills, plus the uptake via ingestion, any dietary items, and that dietary uptake is dependent on factors like the ingestion rate and assimilation efficiency. And then on the efflux side, it's the efflux from water, so the loss of uptake from an aqueous exposure and a dietary exposure. So you put together those, the blue parts of the equation and the green parts of the equation, and you can then estimate the concentration at steady state. And we can define those constants or those variables using exposures from the dissolved phase to generate these kind of these curves, and then also pulse chase experiments from dietary items. So we can then define each one of those terms to go into the experiment quite nicely under a range of different conditions. So we did a series of exposures to both cesium and strontium on these curves, again, time on the bottom and concentration in the animal on the y-axis. Cesium is in the red squares and strontium 85 is in the blue triangles. So strontium is a calcium and magnesium analog. It tends to go to a lot of the hard structure like shells in the case of crabs, whereas cesium is very much a potassium analog, so we'll go to a lot of soft tissues. But we found that after 3 weeks, so on the left-hand side, the A curve here, after 3 weeks, there was no equilibrium found. Sorry, that's 2 weeks of exposure, 14 days of exposure, no equilibrium. So those curves didn't flatten off. But you can also see that the data here are 8 replicates, 8 individuals contained in their own individual exposure chambers. So not sharing water, they're 8 true replicates, plus or minus the 95% confidence interval. And you can see that we're able to get some really nicely agreeing data from an individual to individual basis. The second curve there is the loss rate. So this is how quickly the animals, if we put them into clean water, how quickly they remove cesium and strontium from the bodies. And you can see that there's a little bit, after 3 weeks of depuration, there's a little bit more retained of strontium than there is of cesium, but the loss rates are pretty similar. And then the third one there under C is the dietary exposure. So this is where we prepared some radio-labeled food, fed it to the animals, and then analyzed them hourly or daily over a period of 21 days to work out how quickly they assimilated the metal, and then also how quickly they were able to get rid of it. And sure enough, the rate curves down here, the slower loss rate curves are very similar, whether it was an aqueous or a dietary exposure, which is what we would have expected, in that when the contaminants are inside the body uses very similar processes to get rid of the contaminants under normal conditions. And I say under normal conditions because we then wanted to do a longer study, a longer exposure, to see, could we find equilibrium? Is there a point where we can get these animals and the curves plateauing? So we did a up to 8 to 15 week exposure period, so a very long exposure to an aqueous cesium and strontium. And these triangles here are for an individual exposed to strontium 85. And you can see that it's actually a very linear uptake rate. The red triangles here are for another separate individual who was following along the uptake rate very similarly, and then at this point molted. So it shed its shell in order to grow. Invertebrates will shed their shell, regrow another one that's slightly bigger in order to grow larger. And you can actually see, hopefully on the picture there, that Crabs are amazing in that they shed the majority of, well, all of their hard shell, but then also they shed their gills. I don't know how one goes through losing soft tissues like that. It must be amazingly painful. If crabs could feel pain, I guess they don't, because we don't tend to require ethics approval to work with crabs. But interestingly, yeah, they shed their gills, and in this case, the amount of strontium that went into the body significantly increased. So the rate at which the animals accumulating strontium was huge. And actually, we had 2 animals that molted on exactly the same day, and they both followed exactly the same profile. I'm just showing one here for simplicity. But after 46 days, which is when we had to, or 46 days post-molt, which is when we had to terminate the experiment, the uptake rates were still different. So the uptake rate of the malted crab was still significantly different from the non-malted crab. It was accumulating strontium more quickly. With exposure to cesium, again, after this is now 15 weeks of exposure and individuals accumulation, you can see that there was a slight tapering off of the rate, but it still didn't reach equilibrium. It didn't plateau and find its equilibrium. But interestingly, again, in the red here is another individual that was following the uptake rate of the green one very similarly, but then started to increase its accumulation. And then at this point, molted. And for the next 18 days, actively started to lose cesium. This is the first report to our knowledge of any animal actually depurating, so losing cesium while it's still being exposed. And this is, even if you account for the fact that it's increasing in mass, that still doesn't account. So your per gram, your gram is going up, but that still doesn't account for the fact that it still is actually losing concentration. And then interestingly, after 18 days, the uptake rates were then comparable to the intermolt crabs. So these techniques give us a really nice way of understanding these, or following the physiological changes and how contaminants behave during these physiological changes that the animals are undergoing. Under depuration was a similar kind of profile to what we had seen before, but again, we had a few under depuration. So this one featured in the red circle that molted, and for 20 days lost cesium at a greater rate than its intermolt cousin who was in the tank beside it. So yeah, it gives us some really nice insights into what's going on. From the imaging perspective of this study, we had a look to see where cesium was within the animal, and we were able to image the claw. So this is a cross-section through the crab's claw. On the left-hand side, this is one that after it had been exposed to radiocesium in solution for 15 weeks, and you can start to see it's not homogeneous. It's certainly within different muscle structures. So we're even getting partitioning of cesium to different muscle structures. The one on the right here is cesium in the claw, albeit that there's a not as good a signal to noise ratio. The radioactivity is giving us everywhere where the darker blue is where the radiation is. But this was cesium in the claw tissue 6 hours after ingestion. So cesium was accumulated from food. We gave the animal cesium contaminated food. 6 hours later, euthanized it and then did some sections of it, but it was labile enough that it was bioaccumulated through the gut, transported through the circulatory system, all the way through to the exterior muscle tissue within 6 hours. So some pretty fast internal kinetics. We also thought that we had found radiocesium in the crab's gonad, which would have implications because this is a radioisotope. So implications for dosing the main reproductive organ of the animal. But we did some more further imaging on the Australian synchrotron using X-ray fluorescence microscopy, which allows us to gives us X-rays that provide us with elemental maps on a really high resolution scale. So this organ that we're looking at, the gonad, is 0.13 millimeters, so 130 microns in length. So pretty fine definition that we got. And we could see that zinc and copper were certainly flowing around this organ. Strontium and calcium were not there, but they were there in the shell. So this is the strontium and calcium in the top. And then high metabolic turnover as defined by sulfur and potassium. So the synchrotron mapping showed us that while we couldn't see cesium from the synchrotron XFM maps, certainly there were high concentrations of potassium in this organ. However, when we looked at it on a organ basis, the gonad here for cesium wasn't significantly higher in radiocesium than it was for any of the other organs. But interestingly, it was for radiostrontium. So of all the organs that we dissected and quantified the amount of radioactivity in each organ, the gonad had the highest concentration of radiostrontium. Strontium-90, which is the predominant radionuclide that comes from nuclear fission. So when you separate uranium in a nuclear reactor, you end up, some of the daughter products or the fission products that you get are cesium and strontium-90. And strontium-90 is a pure beta emitter, so a high energy electron basically that can generate a pretty high dose. So if we don't yet know what the significance is of having a lot of strontium-90 within the gonad, but it potentially would dose that organ to a point where it may not function as a non-irradiated organ may do. So there's still further work to go on there. And just to tie this bit of work together, we took the biodynamic model that we had at the beginning that I introduced, put all of the different factors in, and then also made some assumptions about the fact that the animal didn't grow. Under our model assumptions, we used data from Japan to give us concentrations of cesium and strontium in the water and in sediment, and then assume that the dietary pathway of crabs will have included a significant sediment component because they're scurrying around the sediment. And we were then able to model to find that before the accident, the cesium, predominant pathway of cesium would have been via the diet. Soon after the accident, for animals quite close to the plant, water would have significantly dominated because there was a lot of radiocesium that went into the water. And then data from 1 to 3 years after the accident suggested that the balance was then back to the dietary phase. For radiostrontium, it's quite different and was different from what we had hypothesized, but water was a significant component of the uptake. So this is the percent contribution from each source at the different time points, and certainly water just after the accident accounted for nearly 90% of that's what we modeled. The water would have, the aqueous phase would have accounted for nearly 90% of the strontium-90 uptake, and even 1 to 3 years after the accident, still about 80%. So this sort of information then really helps to understand that risk, the response to a specific accident, and then also the cleanup afterwards if the contaminant is going to be predominantly coming from the sediment source versus the water source, and what you can potentially do in harbors or other waterways to clean up those particular contaminants. So that was the story that we went on through there. And I'm going to just spend the next few minutes running through in a little bit more brief some of the other studies that we've done using radiotracers. So the next one is looking at the potential for insoluble nanoparticles to accumulate through freshwater food chains. And this is work done with colleagues at CSIRO with Lisa Golding. Cerium dioxide is a very insoluble nanoparticle that's added or had been added in the past to diesel fuel. And then you've got a potential route to the environment via the combustion of diesel fuel, and then run off into waterways. So we wanted to ask the question of whether or not these particular insoluble particles would be accumulated within a simplified freshwater food chain. So we took cerium dioxide particles and we added them into the research reactor out at ANSTO, made them radioactive, and we then carefully, but formulated them and mixed them with freshwater algae, and then fed the algae to snails, and then fed snails to prawns. And the series of images I've got here is watching how the just after ingestion, the radioactive snails are within the animal's mouth and stomach, as one would have expected. 6 hours later, the radioactivity is confined to the stomach and the start of the gastrointestinal tract. And then 24 hours later, where the biokinetics had shown us that the animal had depurated 97%, lost 97% of the activity that it had ingested. The only trace activity was found in the mid-gastrointestinal tract. So this worked really nicely to actually show that these insoluble nanoparticles, and from memory, if I remember rightly, they were 50 to 100 nanometers diameter as the primary particle size, but they did not bioaccumulate into the animal tissues. Otherwise, we would have seen uptake into the hepatopancreas or other tissues. In this case, we were able to show that the particles are passing really nicely through the animal. We're in the process of working with the International Atomic Energy Agency and other collaborators to develop microplastic radiotracers. So where we can do the similar kind of thing with microplastics at this stage to again understand, is there any true bioaccumulation of microplastic particles through the guts at environmentally relevant exposure concentrations. Some of the other work that I've done with colleagues up at Griffith University in the Smart Water Research Centre, so this is with Dr. Chantal Langto, has been on looking at amphibians and understanding metals that may be liberated from coal mining, as well as other heavy industry, and their potential accumulation into native amphibians. So we took inorganic salts of selenium, sodium selenite and sodium selenate, added them into the reactor, made them to selenium-75 as radioactive, and then conducted a series of aqueous exposures into the animals, and had found that the selenium-4 accumulates more quickly than selenium-6 does, but that both were depurated over time to a very similar degree. This was really interesting, suggesting that there's then a conversion to another, either conversion of one inorganic form to another, or conversion of both inorganic forms to an organic form, selenocysteine, methionine. So we had done some extra work at the Australian synchrotron to show that actually uptake into different organs was very much converted into the organic form of selenium almost exclusively. We also did some nice work with the radiotracers, following how selenium changed within the body through metamorphosis, I should say. So the first panel A on the top left side is the cross-section of the animal exposed to selenium for 7 days, put into clean water for 3 days, and then euthanized and sectioned. And then the B, C, and D panels are where the animals were still in depuration, so in clean water, taken out at different stages of metamorphosis to understand where the selenium had gone within the animal. So for things like the limbs, the arms and the legs, which weren't present during the exposure period at A here, selenium was mobile enough within the body that it then was partitioned out to those other organs during the depuration phase. We also saw something similar within the eye lens as well. Selenium is an essential micronutrient and is required for particular eye development. So in the lens, as we can see, and the retina, but too much selenium in the eyes can cause the formation of cataracts and other issues that then can impair vision, which then you've got potential for population level impacts. So if you're losing your vision, you've got issues finding a mate, you've got issues evading predators and finding food. So all potential population impacts. We didn't necessarily, in this case, quantify the impacts from selenium. It was more just about understanding how selenium changes in terms of its internal distribution as the animal goes through metamorphosis. Chantal Langteau is currently at ANSTO, or will be here this next week, and we're redoing similar studies but using methyl mercury. So we'll be doing a series of dietary studies using methyl mercury and understanding how the biodistribution of methyl mercury changes over time. We've also managed to get some really nice elemental maps of different metals within the animal tissues from the Australian synchrotron on the XFM beamline. So this was a series of tadpoles that were exposed to selenium in solution, and then we imaged them and were able to find that, yes, sure enough, selenium in green versus zinc in red and sulfur in blue accumulate very much in the iris and the eye lens, whereas we're also finding selenium in the brain, in the liver, and interestingly in places like the lungs. So while this animal is still breathing, still in the water, using its gills to extract oxygen from the water, the selenium is labile enough internally that it was being petitioned to the developing lungs, as well as going to the notochord, so the precursor to the spine of the frog. So we're getting some really cool images and understanding about distribution using our nuclear techniques of radio tracing and the Australian synchrotron. Brad, how much time would you like me to have a little bit more, maybe another 5 minutes?
Yeah, if you could wrap up in 5 minutes, that'd be brilliant, Tom. Thank you.
Okay. I might just jump ahead a couple here in the interests of time. So bear with me, everybody. Hopefully that works. And we'll come along to, excuse me, I'm just going to jump through some of these. Okay, just go on to some recent work by PhD student Sarah McDonald down at the University of Melbourne, looking at urban runoff during stormwater events. So metal accumulation in freshwater species. Sarah's asking the question, do metal pollutants present in pulsed storm events pose any detrimental effects to local freshwater species? And the idea was to try and use radioisotope tracers to examine the biokinetics, organ distribution, and potential biological effects of metals during these particular pulses. And ultimately looking at whether or not the current guidelines are protective for pulsed events where one can have short duration but high concentration of exposure. So Sarah came up to Lucas Heights to ANSTO to do some work with us and did some exposures with dissolved zinc, and the 1.24 milligrams per litre concentration of zinc is high for normal flow, but certainly has been detected in big stormwater events. And so she did some exposures that were pulsed and then analysed the animals after each pulse, as we can see coming up through the exposures. And it was actually a relatively linear accumulation, regardless of the fact that there were breaks in the exposures. Then the animals were depurated, and after the 3 pulses and after the end of the depuration were imaged via autoradiography to understand where the contaminants go in the body. And sure enough, zinc in the gills, but then after 8 days of depuration, or 7 days of depuration, zinc was still present in the gills, but also in the antennal gland, which is an organ for urea production and so urine loss, so potentially an excretion organ. She also did a similar thing with cadmium, and interestingly it followed a different response. So the accumulation pattern was quite rapid after the first pulse, less uptake rate for the second, but then almost no uptake, no significant uptake in the third pulse. So quite a different response there from the animal relative to just a continual response, continual exposure. And the depuration profile was pretty much non-existent. These animals retained cadmium quite well, unfortunately, and even after 7 days hadn't really depurated anything of any significance. From an imaging perspective, Sarah was able to look at the gills and the hepatopancreas as to the predominant sites of uptake of cadmium. And then at the end of the depuration period, it was a lot of cadmium was then in the hepatopancreas, so the digestive organ of the animal, where it's likely going to be stored for a long time and not been able to get rid of. Sarah had come back as well to look at doing some exposures to blue spot gobies, and this was via aqueous sources and dietary sources. And interestingly, for an aqueous source where she exposed again at the quite high concentrations of cadmium and zinc, we found no detectable accumulation. We've had some work done with colleagues at the IAEA labs in Monaco and Europe who have done something very similar with their marine gobies and have again found no accumulation of cadmium and zinc even when they're exposed to high concentrations in the water. So we're looking into that a little bit more as to these specific fish, as to why they don't accumulate via aqueous sources. But there was uptake via dietary sources, and again, she did look at single feeding versus multiple feeds, looking at that sort of pulsed effect, and then where the radionuclides and therefore the metals went within the body. Cadmium, regardless of whether it was a multiple or a single exposure, ended up within the gastrointestinal tract, and so it was very much retained where it was exposed to, whereas zinc ended up within the carcass of the body. And there was also an induction of metallothionein after exposure, single and multiple exposures to cadmium. I believe this was cadmium and zinc, so there was some organ response as well. And just going to finish providing you some information about the work that we're currently doing, looking at decommissioning offshore oil and gas structures. The current legislation says that you have to remove all of your infrastructure once you've finished producing. So that's all of the floating topside infrastructure, as well as any of the pipelines and wellheads. It's increasingly becoming questioned as to whether or not certain pipelines can be left in place, as they do offer a nice reef for habitat to grow on, and there's been some really good work saying that actually that artificial reef can be very productive, especially for fisheries, but that we need to have some understanding about the contaminants. So there are naturally occurring radioactive materials, norm, and mercury that can accumulate in scales, kind of like cholesterol on the inside of an artery, on the inside of these pipes. So it's important to understand what contaminants are there if certain infrastructure is to be left on the bottom of the ocean. There's a huge economic liability for removing all of this infrastructure over the next 50 years or so, and even just examples of 1.5 kilometers of pipe needing to be removed at a cost of over $4 million US in total to be removed and cleaned. So the work that we're doing at ANSTO, excuse that pop up there, that should have come last, but we're working through a process to characterize pipeline scale, conduct solubility tests to understand the potential for any contaminants, so the naturally occurring radionuclides and or mercury to leach into seawater and also into dilute acid, and then conduct bioaccumulation tests as well to understand whether or not any of the contaminants present in these pipeline scales end up within the bodies of the animals that are ingesting them. And then we've got several research projects underway to understand in a lot more finer detail the potential impacts of naturally occurring radioactive materials and mercury. So we've got some great work hopefully coming out in the next couple of years in that space. Hopefully with that, and thank you for bearing with me through that, and apologies for having to skip through some of the format that I thought that I would be able to cover. But hopefully I've given you a bit of a flavour for where nuclear techniques give us some really sensitive tools to understand the uptake of nutrients and contaminants. I didn't go into the nutrient side here, but you got some pictures of some colleagues doing some work with crop plants. But it's a really fun and interesting technique, and obviously we do it very safely as well, working with radiation. You can imagine, I didn't mention about working with mercury. We've made it radioactive and then methylated it. So working with radioactive methylmercury, there's a lot of safety paperwork behind that too. And thank you to all of the people that are involved in the work. This is certainly a group effort with multiple students and collaborators. So it's a wonderful team of people to work with. Thanks for your time.